Hello, it's Avinash here. Today topic is about vectors, which special deals with that of your cloning or what we call multiplication of your desired gene. Before going to the cloning, uh, I would like to be introduce techniques like what we call PCR, polymerase chain reaction. We all know about that. So the question that arises in our mind that why we do PCR, and with along that PCR we have to be continue the method of cloning. The two basic reasons behind this is that the first one, PCR has a limitation of the size that up to which it can clone or multiply the DNA sequence, which range lies in between 4 kilobase up to 40 kilobase, and those eukaryotic genes having a size which is usually or what we call normally greater than that of the size of the PCR containing. But in case of cloning, the size we can go up to megabase pair, and that's a positive point. Second thing is that in PCR we have to know the sequence of what you call the primer, the oligonucleotide sequence, which with our our desired gene DNA has to be anal, and to know the sequence of that primer, we have to go through cloning. If a DNA is not yet cloned, then we cannot run a PCR because we have no idea about the sequence of that primer of that particular gene. So this is the two reasons why we need to be continue our cloning technology with that of your PCR. No doubt PCR is a very well known technique and is much efficient than that of your cloning, but we cannot leave this cloning technology. So let's begin the cloning technology. Only the important I'm gonna read right now. So E. coli, you can see here, is the one of the bacteria which is it is the most common bacteria, and you know all that it's normal structure. So this E. coli is very commonly used because it's found in our gut intestine, and it's easy to grow, simple organism, a rapid doubling, is easy to understand, having many molecular work has been done, complete genome sequences, and etc etc. So this brings a beneficial point to work on this particular E. coli bacteria. The bacterial cells were able to exchange the genetic material, so the experiments of Liedenberg and Totem to early demonstrated the transfer of genetic information through the bacterial conjugation. The genes can exist on a circular piece of DNA that replicates independently. That is your plasmid I'm talking about here. They can be double stranded, replicate that of your own. That's of their own. And there's some F episome that carry only the fertility gene, etc. Then HFR, dragging the rest of the genome behind them. Aptex, where the other genes are transferred very efficiently from the bacterium to the next because the length of the transferred DNA is short enough that it can move across the connection between the two bacterial cells before the virus break. And anything else? Okay, so a vector that is used predominantly for producing the DNA fragment is often referred to as your cloning vector, while if it is used for expressing a gene contained within the cloning DNA, it is called your expression vector. Then this is some of the vector with their main use and their maximum insert site. As you can see, in case of plasmid like your PBR32, PUC 18, the maximum size is up to 10 to 20 kilobase, which is similar to that of your PCR approximately. In case of lambda insertion, This is important because it's used to construct the complementary DNA libraries. Size is also in between this range. Example, you can see Cosmids having a construction of genomic library, also having a capacity up to 34 kilobase. That's great. Then another one is M13, which is about 8 to 9 kilobase. That's not so quite impressive, but there's some advantages because it helps in sequencing studies. Then plasmid YSC, the East artificial chromosome. I'm talking about this because it can carry a size up to 1 to 2 megabase pair. That's very interesting. Then nothing else. So plasmid are widely distributed throughout the prokaryotic in a range in size from approximately 15 to 300 kilobase per basic common things. I should not read out all those things. Plasmids are often described as being either relaxed or strained on the basis of the number of copies of the plasmid that are maintained with the cell. So the plasmid that found inside the bacteria are up to two types. One is a relaxed plasmid and the one is a stringent plasmid. In case of stringent plasmid, it's present only one or two, and whereas the relaxed plasmid which are in multiple numbers. So what does the function? Well, the relaxed plasmid replicate using a host derived protein, yes, but while the stringent plasmid encode the protein factors that are necessary for their own replication. That's the difference. Anything else? Here's written as a naming techniques, etc. So one more thing is that col E1 is a 6646 base pair closed circular DNA molecule that encode a bacteriocin. Colicin E1, that is a product of CEA gene and resistance gene that allows the host bacterium to escape from the effects of the bacteriocin, is your IMM gene. That means the IMM gene, which is a resistance gene to a particular bacteriotoxin like your bacteriocin, which is produce colicin E1 from the gene that is your CEA gene. So a drug resistant gene encodes for a protein that inherits with the action of colicin by inhibiting the ability to form a canal through a bacterial membrane. So different. So unlike the bacterial origin of replication OAC, however, the replication col E1 proceeds in one direction only, etc. Okay, this is important also. Unlike the bacterial origin of replication like OAC, the replication of col E1 proceeds in a single direction. Replication of col E1 I'm talking about. That's also confusing me. Here written, through the complementary base pairing, RNase2 can be bind to one of the DNA strand only, and hence the replication of the col E1 plasmid is unidirectional. That's the reason. Okay, that's fine. Two non-translated RNA molecules termed as the RNAi and RNAi and the protein product of the ROP gene control the replication of the replication process. That's important. The RNAi and the RNAi and some protein product of the ROP gene control the replication process. So how? Let's see. Well, the control of replication process is achieved by another non-translated RNA molecule. RNAi is complementary to the 5' end of RNA2. Getting this point? And the RNA duplex formed between the RNA2 and RNAi cannot serve as a replication primer. RNAi is a relatively short-lived species, and consequently the col E1 plasmid is maintained at a level about 15 to 20 copies per cell. The interaction between the RNAi and RNA2 is stabilized by ROP protein. That's the thing. We're talking about the chloramphenicol. This chloramphenicol increases the plasmid number, and this is done by decreasing. Here it's written as the chromosomal DNA replication is halted, but the COL-E1, that's product the chloramphenicol based on the plasmid continue to replicate and accumulate high levels of copies per cell. That means the plasmid per cell increases in presence of that your chromamphenicol. There's some different plasmid PBR322. This is PBR322 carries the COL-E1 replication origin ROP gene to ensure the reasonably high plasmid copy number. As you can see here, the COL-E1 replication origin and ROP genes to ensure reasonably high plasmid copy number. So this brings the copy number to be increases inside the cell. That means our desired gene get increased or multiplied. That is up to 200 fold. That's interesting. And this is done by the chromamphenicol amplification. As I told you, this chromamphenicol cause what? This reduces or halts the DNA replication, and thereby it allow the plasmid to continue replicated and accumulate at high level inside the cell. Then coming to the antibiotic resistance, this is a picture which shows to possess some ampicillin and tetracycline, which is resistant to those kind of drugs because the protease enzyme which can be to protect those bacteria to survive in that medium. And here's some also shown here that the PST, PBU, these are some restriction enzyme which cause cleavage of a particular site in the gene. So if you just see a little bit carefully in the diagram where it is, okay, here the PST, you can see the PST, the BAMH1, tetracycline, ampicillin is a site respectively which get clipped out in presence of two restriction enzyme. And then let me see anything else. No, in other words, the insertion of foreign DNA fragments into antibiotic resistant gene. No, that's not so important. Okay, here is the important thing. One is that we want our desired gene to get inside where? Inside the plasmid, the vector, which is will multiplied inside their host body. So to get an insert DNA fragment, we have to isolate those particular segments via using enzyme like this BAMH1, which can cut those sites, and then we have to apply those BAM to cut in the plasmid. And here the BAMH1 causing a cleavage in between tetracycline resistance gene region. So once this inserted gene get accumulated inside the plasmid at that particular site of tetracycline, that means the tetracycline gene is becomes non-functional. So it's like an inhibited or what we call mutated, something that means it will not work because the sequence get disturbed in presence of another insert gene. So we can filter out that recombinant type by placing it in a colony containing plasmid with the ampicillin or agar based, because the ampicillin is present but not the tetracycline. So once we get that, then we can replicate the colony with non-recombinant plasmid but with an ampicillin and tetracycline, because we know that tetracycline resistance only will be there having a non-recombinant type, but here the colony contain recombinant plasmid will be get died, and we then having the sample from where it's taken from the source, we can consider the source as your recombinant plasmid containing host. Hope it's not that complicated to you understand. Here is a high copy number, and mutation within the origin of replication produce 500 to 600 copies of plasmid per cell without the need for chromophenical amplification. The mutation in a G to A branch, so different things, and this is very very many more things. So colorless, non-color. My God.